Hi everyone, Dr. Hinkey here with the video lecture for Chapter 3, the chemistry of organic molecules or biological macromolecules. So this will get us through our week 2 chapters, 2 and 3. As I said, the pace will slow down a little bit after week 1. But this week was loaded with chemistry, which might be new to most of you, although you're probably familiar with the concepts, especially when you got to this chapter and said carbohydrates, lipids, proteins, nucleic acids. I've heard of those. This is nutrition, but our nutrition comes from the things that we eat, and the things that we eat, we're living things. So they are made up of the same molecules that we're made up out of, and the things that we need to eat we get from other organisms. So the first thing we'll talk about is organic molecules. So why do we call them organic? And what's the difference between organic chemistry and inorganic chemistry? If any of you are going to go on and take more chemistry. When we talk about organic molecules, we're talking about molecules that contain both carbon and hydrogen atoms. So our carbons are bonded to some hydrogens. It's not just carbon. Carbon dioxide has carbon. Cyanide has carbon, but neither of those are organic molecules because they don't have hydrogens attached to that carbon. So that's what's going to differentiate it. And in organic chemistry, all the molecules that are studied have carbons, either single carbon or carbons in a ring. So a circle of carbons, each connected to each other with hydrogens and other elements attached to those, or long chains of carbons with hydrogens and other elements attached to those. So that's organic chemistry. That's what makes something organic. And just a word of caution, there is a different commercial or agricultural use of the term organic compared to chemistry. So organic farming and organic produce have certain requirements that they need to meet to be labeled as organic in the grocery store for sale, for consumer use. If you want to be a certified organic gardener, there are certain things that you have to do, and one is not to use pesticides or synthetic fertilizers, and both of those are organic molecules. So there are a lot of organic pesticides. So organic in the chemistry sense doesn't mean it's good for you or healthy. So pesticides, I know a common thing I've heard from mosquito control places here is say, well, I don't want any harmful chemicals. Oh, our pesticides are all organic. Those are some of the most toxic pesticides out there because of those long chains of carbons and hydrogens, petroleum hydrocarbons. Those are organic. So doesn't necessarily mean healthy when we're talking about chemistry. It just means that we have carbons and hydrogens attached. So we have 4 classes of our biological macromolecules. These are present in all living organisms: carbohydrates, lipids, proteins, and nucleic acids. So these are the 4 big categories, and we have specific molecules with important and specific roles within each of these. So their functions are varied, lots and lots, and we'll look at those. So our organic molecules, those carbons and hydrogens, even in petroleum hydrocarbons, they come from or are derived from living cells. So petroleum hydrocarbons come from long dead dinosaurs and ferns and plants that have been dead and buried and decomposed, losing their oxygens, leaving behind long chains of those carbons and hydrogens. Inorganic, if you don't see a C and an H in the chemical formula, it's inorganic. So we're interested in these 4 classes of biological macromolecules because these are the components of our cells: carbs, lipids, proteins, nucleic acids. So they're biomolecules, biological macromolecules. You'll sometimes see them referred to macro because These can become very big. They're not all big, but they can be big. And these usually consist of many repeating units. That's why they can be small, but a lot of times in their final structural form, the form that they function in, they can be very, very large. And they're usually, for those 4 classes, they're made of repeating units. So each repeating unit is called a monomer, one single unit. So if you think about a house made out of bricks, the monomer would be 1 brick. And then I can stack those bricks into different shapes. And one of those is lots and lots and lots of bricks I can stack into a house. The house would be a polymer. But using that same example, think of all of the different structures I could make using that monomer of bricks. All the same building blocks, but I put them together in different ways and I can get lots of different things. I can get a brick wall, I can get a brick house, I can get a brick sidewalk, I can get a barn, I can get all sorts of things made out of bricks. So in our biological macromolecules, an example would be that amino acids are the monomer. That's my single building block, my brick, and they're joined together to form proteins, a polymer. So really, we form them together to get big, long chains of amino acids that then fold and twist into the shape to become a protein. But the big, long molecule, the repeating units, that is my polymer. Lipids aren't technically polymers because they have 2 different subunits. So we put 2 different things together, but then those subunits all come together to form big lipid molecules. So lipids are sort of our odd guy out here because they don't quite follow the same pattern as the others to some extent. But they sort of a little bit do. So we'll look more closely at these and what their monomers are. So carbohydrates, my building block or my brick, or my single Lego. I like the Lego example because it's really perfect. I can have 1 Lego as my monomer. I can put the same Lego together in lots and lots of different ways, get 2 different structures using the same building block. Or I can take lots of different Legos and put them together in really complex structures. I can get a house. I can get a sea turtle. That's at the aquarium. If anybody hasn't seen that, they had lots of Legos on display. So this column here, this second column, my monomers are my single Lego. What I'm going to build with them is going to be my polymer, lots of those bricks stacked together. And then within my cell, I can put combinations of all of these to get lots of stuff. So carbohydrates, my single building block is a monosaccharide. Mono is 1, and saccharide is sugar. So 1 sugar. If I have 1 sugar, that is my monomer. This is a carbohydrate. Simple sugars are carbohydrates. I put lots and lots and lots of them together. I can put them together one way and get a funky, twisted tree trunk. I can put them together another way and get a wall. I can put them together lots of different ways to end up with different shapes with different functions. And those are polysaccharides. So poly means many sugars. Polysaccharides are made of sugar, but chemically they are distinct. They have distinct and different chemical properties from sugar. So a starch is made of sugars, but it's not a sugar. But both of these are carbohydrates. Lipids are my funky group. We'll look more closely at these. My lipids of primary concern have 2 subunits, a glycerol and then fatty acid tails. And we'll look more closely at that, see what that looks like in a minute. And when I put these together, depending on what I'm putting together, I get structural components of a cell, but we're not going to really call that a polymer because they're put together in weird ways. We'll see that in a bit. Proteins. This is a great example. My protein, my building block, is an amino acid. So here's one. There are 20 amino acids that we use in our body. There's 20 amino acids, so we can think of that kind of like the alphabet. I put letters together to get words. I can get short words, long words, all kind of words, all kind of different orders. 26 different letters, I can put them together in lots of different ways. My proteins, I can put those 20 different amino acids together in all sorts of different ways and different orders, longer, shorter. But when I join amino acids together, I join them with what's called a peptide bond. And I join the nitrogen from one amino acid with a hydrogen in the adjacent amino acid. That is called a peptide bond. You don't need to know how that bond is formed, but you need to know that's why this is called a peptide. And a polypeptide is a big long chain of amino acids because there are many of those peptide bonds. In this case, my amino acid is not a protein. My chain of amino acids, let's say this is an amino acid and here's my chain. My chain of amino acids would be called a polypeptide. It's still not a protein. This one's on its way to becoming a protein, but these big long chains of amino acids have to bend and twist and kind of fold back on themselves to get a very specific shape. And it's not until they've taken on that shape that we would call them a protein, because the protein, the shape of the molecule, is very critical to its function. So until it has that shape, it can't function as a protein, can't do its job. But the polypeptide is the polymer. And then my last biomolecule that we're going to talk about are nucleic acids. The subunit or monomer of a nucleic acid is a nucleotide. And my nucleotides would be this brick, but this brick would be built of a sugar attached to a nitrogen base attached to a phosphate. So there are three components to this one brick. So just like my Legos can have bigger or smaller pieces. And this I'm going to put together to form my nucleotides are going to be put together to form nucleic acids like DNA or RNA. And I also have a nucleotide that I'm going to use by itself called ATP, which is an energy molecule. DNA and RNA are information-carrying molecules. So that idea of monomers, polymers, this is, if you get this idea and the names of those monomers, it's pretty easy to figure out how these guys work. So how do I get two monomers to join? Well, I get them to join through a process that's called a dehydration reaction. So if I want to join molecules, what does that name suggest? Dehydration. I remove a water. So dehydration reaction, sometimes this is called dehydration synthesis. Remember, if I synthesize something, I'm building it. So synthesis, dehydration synthesis, I'm building a molecule. Or it's also referred to, I think in the lab manual, as a condensation reaction. And in condensation, water comes out of the air. So I'm removing a water for all of these names: condensation reaction, dehydration synthesis. I'm going to remove a water. I'm going to remove a hydrogen from one of my monomers and a hydroxide from the other. And those two are going to come together to form a water that's going to leave. And where that hydrogen and hydroxide came off of my monomer, I leave some atoms looking for a partner to fill their valence electron. And so they're going to come together and form a chemical bond between two monomers. So that would be a dimer, two, and then I keep doing that to get big long chains, a polymer. So the converse is true if I want to break down a big polymer to a small one. So for example, why would I want to do that? When I eat too much sugar, if I have those Oreo cookies for breakfast and I have lots of sugar in my bloodstream, I don't need that much. So my body is going to store some of that by capturing it, taking it out of my blood, and storing it in my liver is glycogen. So glycogen, so what it's going to do is remove those sugars, those monomers, and it's going to package them together into a big giant molecule. And then as my blood sugar falls throughout the day, it's going to gradually release those sugars. Well, they're in a big giant package and a polysaccharide, not a sugar, but I can break off one sugar at a time and release that into the bloodstream. And I do that through the process of hydrolysis. So lysis, if I lyse something, I break it apart. Hydro is water. I'm going to break a big molecule into smaller pieces using a water molecule. So here is my monomer. This top picture is an example of synthesis. I have one monomer. I have another monomer. So these all contain carbon and hydrogen, and all of these contain oxygen, but not all organic molecules contain oxygen, but all of my biological macromolecules do. And so what I'm going to do is, here's a carbohydrate monomer, a monosaccharide, has carbon, hydrogen, and oxygen. Here's another monomer with carbon, hydrogen, and oxygen. I'm going to remove one hydrogen from this monomer, one hydroxide from that. So I'm going to remove a water molecule, and that leaves this looking for some place to hold on to, and this looking, I'm looking to form a bond, and there. So now I have, my monomers have been joined, and then I keep doing that over and over to get a polymer, my polysaccharide in my example. Here on the bottom, I have the opposite. I have a big long molecule, lots of monomers joined together, and I want to break it down into smaller particles. So what I'm going to do is I'm going to add a water, and by this water coming in here, the water is going to split and a hydroxide is going to come over to this monomer. A hydrogen is going to come over. A hydrogen will come to this monomer, and I'll split them. I separate them out. So hydrolysis, hydro, water, lysis, to break it apart. So hydrolysis breaks big molecules into small. Dehydration synthesis takes small molecules and joins them together to a big one. For synthesis and degradation to occur, I need specialized molecules called enzymes. So metabolism is all the chemical reactions in a cell. Every chemical reaction in the cell is needed at exactly the time it's needed, at the rate it's needed, when it's needed, and so it has to be carried out fast enough to meet the cell's needs. Chemical reactions take place when two elements come close enough to one another that those valence electrons can interact. That wouldn't happen fast enough to sustain life. And so our cells use enzymes to speed up chemical reactions. So enzymes are called catalysts. A catalyst is something that makes something happen faster. So enzymes speed up chemical reactions. They're not used up in the reaction. They're not part of the reaction. They're not changed by it. They just bring two things together. So you can think of it as a yenta, as a matchmaker. A matchmaker brings two people together. They get together, they get married, the yenta leaves. The yenta is still a yenta. She's going to go do that to more couples. So that's what enzymes do. Some enzymes break up couples but the yenta is not part of the marriage. She doesn't enter into the marriage. She's not involved in it. She just gets them together and moves on. Right? So she's not changed by it. She's not part of it. So same thing with our enzymes. They are either matchmakers or match destroyers. They either join molecules together or break them apart. That's what enzymes, they speed up chemical reactions. So all of these reactions, when we're talking about hydrolysis or synthesis, there's an enzyme involved, and that's going to be coming up in a chapter ahead next week in chapter 6. All right, so let's look specifically at our carbohydrates. or at each class of our macromolecules, are carbohydrates. Kind of tells you, oh, they've got carbs, they've got hydrogens, they also have oxygens. The function, what we use in our body carbohydrates for, primary use is for an energy source. They can also have a structural role. They can be building materials, but they are our body's preferred and best source of energy. Carbohydrates always contain carbon, hydrogen, and oxygen atoms, and only carbon, hydrogen, and oxygen atoms. These are usually in a 1-to-1 ratio. So we saw the chemical formula for glucose, C6H12O6, 1 to 2 to 1. So twice as many hydrogens as carbons or oxygens. We have monosaccharides, single sugars or simple sugars. Disaccharides would be two sugars joined together. These are also considered simple sugars. Like table sugar is a disaccharide. Glucose is a monosaccharide. And we join lots and lots of those together, we get polysaccharides. So a monosaccharide is one, a single sugar or a simple sugar, has a backbone. It's not even a backbone, it's a ring. The backbone, sort of the foundation of this molecule, is between 3 and 7 carbon atoms, and they are in a ring. So there are 3 carbons in a circle. Some examples of this, and they've got hydrogens and oxygens attached. Glucose is our blood sugar. Fructose is the sugar found in fruits, and galactose. Ribose and deoxyribose. Maltose, galactose is the sugar found in milk. Ribose and deoxyribose are sugars that are found in our nucleotides. Those are our monomers of DNA. We'll talk more about deoxyribose and ribose when we get to our nucleic acids. So here's an example. This is my glucose, C6H12O6. You don't really have to know all of where all those carbons and hydrogens are. You can count them if you want, but it's a ring. And in this ring, our carbons are the foundation for this ring, and then the hydrogens and oxygens are connected to those 6 foundational or ring carbons. To help us figure out what's going on in chemical reactions, we sometimes do need to know the orientation of the molecule. And so to do that, we can number the carbons in this ring. And by convention, we always start with the carbon that would be, closest to like the 1 o'clock over here on the right. Or if there was a carbon up here, it would be this one. But we would call that by convention, this is carbon 1. So this is C1, C2, C3, C4, C5, and then in my glucose structure, C6 is actually attached to C5, not in this ring. I've got an oxygen there in the ring. So that's just how we would draw this, and you can see glucose is a 6-carbon sugar, and that ring forms a hexagon. Disaccharides are 2 monosaccharides joined together. So lactose is another milk sugar, is a disaccharide. It's a galactose. That's a milk sugar. Joined together with glucose, that disaccharide is the complete sugar in the milk. Sucrose is table sugar, and that's 1 fructose combined with 1 glucose. So di means 2. Maltose is 2 glucose molecules. And just for any lactose intolerant individuals, since I already mentioned enzymes and we'll be talking about them more, they're really, really important. If you're lactose intolerant, that means that your body doesn't produce the enzyme that breaks down lactose into its monomers, into the monosaccharides, galactose and glucose. So you can't process that, you can't do that chemical reaction, and makes you lactose intolerant because you can't digest the lactose till it's broken down. So this is just an example, since we talked about dehydration, synthesis, and hydrolysis. I can take 2 monosaccharides, remove a water, join them into a disaccharide. That's the arrow going this way. This reaction can go either way, depending on if I remove a water or add a water. If I add a water, it's the hydrolysis reaction, and we take my disaccharide and break it into 2 monosaccharides. All right, so our polysaccharides, we can refer to these as complex carbohydrates. They are chemically distinct from simple sugars. So their chemical reactions, their chemistry, what they do is different than sugars, even though they're built from sugars. So that just comes into play in the lab when we're identifying molecules on why we have separate tests for sugars and starches, because sugars react differently because they're sugars. Starches are made, or polysaccharides are made of sugars, but once we connect them and form those bonds, they react differently in chemical reactions, and so are separate. So we have to have 2 separate tests. So a polysaccharide is a polymer of monosaccharides, and some examples and what they're used for. Starch, this is how I store energy in plants. So I already mentioned glycogen. If I have lots of monosaccharides of glucose in my blood, too many, and I want to store them for later, I can package them into glycogen and then slowly release them later to use. Starch is the way that plants package their excess sugars. Cellulose is another polysaccharide that plants make. They build it from glucose, from simple sugars, and they build the polymer to make their cell walls. It is the most abundant organic molecule on Earth. It's in the cell walls of all plants. And most animals are unable to digest cellulose unless they carry the right gut bacteria to help them out. Chitin is a polysaccharide that is found in the cell walls of fungus and in the exoskeleton of some animals like crabs and clams. Not clams, sorry, like crabs and lobster have chitin. Oh, let me show that. And then peptidoglycan. Peptidoglycan, when you see glyc or gluc, you know it's probably going to be a polysaccharide or something built of sugars. So peptidoglycan, we've got sugars, and this is what the cell walls of bacteria are made of. The peptido part refers to the fact that I have rows and rows of sugar molecules that are, so polysaccharides, they're not sugars, but alternating sugars connected together to make a polysaccharide, and then layers of them are connected by amino acids. So that's where the peptido. So it's got a complex molecule with both amino acids, the building block of proteins, but they're not proteins because they're just amino acids, and my polysaccharide. So just to get an idea, all of those are built with their basic building block of glucose. So you saw that glucose molecule. If I put that glucose together in these nice long chains, lots and lots of these nice long chains, and then I connect those together with chemical bonding, I get cellulose. I get cellulose fibers made from lots and lots and lots of glucose, all bound together into a polysaccharide called cellulose. If I bind that same glucose molecule together, lots and lots of them in this zigzag pattern, I get starch. I can store my excess sugar. So if I look at a potato under the microscope with some iodine on it, iodine will turn the starch black. So that's why I get these big black molecules. Those are starch molecules stored in the cell of a potato. And then glycogen, the way animals store their excess sugar, is this zigzag twisted mess here, but that's all repeating units of glucose, all bound together into a molecule called glycogen. So that's, again, going back to the Lego example I used, I can take those Legos and put them together, that same Lego repeating units, and get all sorts of different shapes and structures. All right, my lipids, this is my messed up group here. These are varied in structure. These are large molecules. They are nonpolar. Just remember, polarity in water means a molecule, one side was slightly negative, the other was slightly positive. These are nonpolar. They are insoluble in water, and we'll look at why that is and talk more about these when we talk about the cell membrane. Lipids are used for long-term energy storage. So my glucose or my sugars that I use for energy, I either have to use immediately in cell respiration to get energy from them, or fairly shortly while they're circulating in the blood. Otherwise, I put it into my liver and glycogen to store it temporarily. That's a few hours long storage. And if I don't use that up in that amount of time, well, I have to make room. I'm going to go and eat another meal. I'm going to have more sugar. I need to make room. So I'm going to go ahead and take that glycogen and I'm going to move it out and I'm going to convert it a little bit. And I am going to turn it into lipids, into fat for long-term energy storage. Fat can also be a structural component of some cells, of all cells. We can use this as insulation for heat retention. We use some lipids for cell-to-cell communication and regulation of cell functions. And we can use lipids for protection. And lipids, this category of lipids, includes fats, which are solids, oils are liquid, phospholipids. These have a very specific structure and function. They have the consistency of oils, but phospholipids. Steroids, so we've probably all seen steroids. These can be chemical messengers. And then waxes. So if we look at the specifics, some examples of each. So fats are long-term energy storage. They're also used for insulation in animals. Examples where we use these are for butter and lard. On our bodies, we would use them for protection. We use fat to protect. Our organs have a layer of fat around them for protection. Oils. This is, plants usually store their long-term energy as oils instead of fats. So they store that in their seeds. And we see that cooking oils, that, canola oil, all those plant-based oils, olive oil. In humans, we have oily sebum would be an example. So that's the oil in our sweat glands that's released from hair follicles. Phospholipids. So our phospholipids, this tells you right here, it's a lipid with a phosphate stuck on it. These are what our plasma membranes, what the cell membrane is made up of. And the cell membrane is the wrapper or the thing that delineates inside the cell from outside. So every one of our cells has this membrane around it. Phospholipids are sometimes used as food additives, but we need phospholipids to keep building those plasma membranes. And the steroids, these are a part of our plasma membrane. So that plasma membrane is made of phospholipids primarily, but it also has proteins embedded in it, and it has steroids in it. Cholesterol is the steroid or the sterol component of animal membranes. Ergosterol is the sterol component of fungus membranes. So these sterols in different types of membranes keep that membrane the same consistency. So it's about the consistency of olive oil. And that makes sure that regardless of temperature, regardless of what's going on, we've got the same consistency in my membrane. It's where we have that sterol there. Sex hormones are steroids. So ergosterol, testosterone, estrogen, progesterone, these are all steroids. And we use these in medicines. And then waxes. Our big one, earwax. We have earwax. It's protective. That prevents bacteria from getting into our ear. It's used to prevent water loss in plants and a lot of animals. So candles, polishes, you're all familiar with waxes. So when we look at some specific forms, our fats and our oils, the long-term energy storage molecules, are triglycerides. There's 3 of something in here, and then glycerides. So our 2 components to our monomers for this are a glycerol and fatty acid tails. So in our triglycerides, we have a glycerol molecule and 3 fatty acid tails. That's the tri. So here's my glycerol. Carbons, hydrogens, some oxygen. There's my glycerol head, and it's attached to 3 long fatty acid tails. So fatty acid tails, they're long chains of carbons with hydrogens attached and some oxygen. So these are my long-term energy storage. Those fatty acid tails we can refer to as either unsaturated or saturated. Unsaturated means sorry, I keep hitting the little mouse thing there and sending us spinning off to new slides. So unsaturated, these tend to be liquid at room temperature. I reversed these on this slide here. They're liquid at room temperature. They have 1 or more double bonds between carbons. So let's come back up here. Well, there's a double bond between carbon. If I have a single bond, I get these nice straight chains that line up nicely. But those double bonds give me a little bend or a little kink. They connect these tightly and I get a kink so I don't get a nice straight molecule. And that's what allows my unsaturated fats to be liquid. They don't line up nicely and form a nice solid repeating unit structure. My saturated fats don't have any double bonds between the carbons, and so they tend to be solid at room temperature like butter and lard. I have my descriptions reversed on there. Whoops. So that's my triglyceride. Glycerol, 3 fatty acid tails. Those are fats and oils. Phospholipids, these are the components of the plasma membrane. They're similar to triglycerides, but they only have 2 fatty acid tails instead of 3. The fatty acid tails on both triglycerides and phospholipids are nonpolar, right? So they don't have a positive or a negative end. They're nonpolar and they're hydrophobic. They do not want to be anywhere near water. And that's true on my triglycerides and on my phospholipids. With fats and oils, you see that all the time. The Dawn dish detergent commercials show us that. They drop oils into the water and we get little round clumps. And the reason those oils ball up is because those hydrophobic tails are trying to get away from the water. So they all turn to face each other, leaving the hydrophilic glycerol head facing outward, and that just forms a ball. In this case, the glycerol head is modified with a phosphate group, so the name phospholipid. So their main function is part of the plasma membrane in all cells. So every cell of any kind of organism has a phospholipid bilayer. If I put phospholipids into water, they will automatically aggregate into that form of those tails, those hydrophobic tails trying to get away from the water. And what that will do is form little balls. So they'll actually form essentially a cell membrane like a bubble, like a balloon, spontaneously in the water. And this lets you kind of see what that looks like. So if they're in water, there's water out here, those tails want to get away from it. As they all curl inward though, they trap water on the inside. Wait, we can't get away from it. And so they'll do a second layer toward this inside. So inside our cell, we have cytoplasm. It's an aqueous solution. It's water-based. Those tails want to get away from it. So our membrane is a phospholipid bilayer because I've got water on either side. These glycerol heads are hydrophilic. They're happy to go interact with that aqueous solution inside the cytoplasm or outside of the cell, and those tails want to get away from it. So they face each other so they don't have to see the water. Steroids, we're not going to talk a lot about these, but just know this is another group of lipids. These are composed of 4 fused carbon rings. They have different functional groups that are attached that give them different abilities. They are a component of the cell membrane, as I mentioned, and they're involved in regulation, in cell regulation and cell signaling, cell messages. So cholesterol, testosterone, estrogen, those 2 are sex hormones. The only difference is the functional group that they have attached. So they have the same carbon skeleton. They look exactly the same, but then we're just going to add one little piece on that's different between the 2 of them, and that makes all their functional difference. Cholesterol is a precursor molecule for a bunch of other steroids. As well as that component of the plasma membrane, can contribute to circulatory disorders because when we eat food high in animal, right? So an animal-based diet, a meat-based diet, well, we're getting all of the cholesterol from those animals. And having excess cholesterol circulating in our bloodstream, it can kind of get sticky and clogs arteries. Here's some example of steroid diversity. This is testosterone. I've got these 4. So when I look at steroids, I've got these 4 fused rings. All of them are the same, 4 fused rings. So here's my cholesterol. A little modification, I get testosterone. A very tiny modification here, and I get estrogen. So we're just going to modify my cholesterol molecule to get these other steroids, many of them. And then my waxes, we don't go into a lot on the chemistry of waxes. We're not going to look at this any more in depth, but long chains of fatty acids connected to carbons that also contain alcohols as a functional group. So that functional group is what's added on to give waxes specific chemical properties. These are always solids at room temperature, waterproof. They're very resistant to degradation, so they're used for protection. So earwax, the cuticle on plants that protects from water loss, beeswax, those are all examples of waxes. Okay, and to our proteins. As we said, proteins are polymers that are made up of amino acids that are linked together by peptide bonds. So that peptide bond is a covalent bond between 2 adjacent amino acids. As much as 50% of the dry weight of most cells consists of proteins. We tend to think of proteins as, structural, strong bones and teeth. But not all animals have strong bones and teeth. As much as 50% of the dry weight of most cells. Your cells don't have bones and teeth, right? So that's because of that job of proteins as enzymes. Every chemical reaction in your cell is catalyzed, helped out by a protein. Your cells have lots and lots and lots of different chemical reactions going on, and so lots of enzymes zipping around doing their jobs. So far, we've identified several 100,000 different proteins. And there's a lot that we haven't identified. So, proteomics is the study of proteins, a really big area in biology. So 2 or more amino acids are building blocks, or our Legos, joined together. We call them peptides. When we get a big long chain of those amino acids, we call it a polypeptide. And we call it a protein once that polypeptide has folded into its particular specific shape. to do the job it's supposed to do. So what are some of those jobs that proteins do? Metabolism, this is the big one right here. Most enzymes are proteins that speed up chemical reactions. And most of the proteins that are active in our cells are enzymes. We do have some proteins that are involved in support, keratin and collagen in hair and nails. We have transport proteins. So we have proteins in our plasma membranes that act as channel or carrier proteins. They act like doorways to transport molecules in and out of the cell. We have hemoglobin, whose job is to transport oxygen around the cell. Hemoglobin is a protein. We use them for defense. Antibodies are proteins, or immunoglobulins are antibodies in our immune system. Those are proteins. Hormones, we just said steroids are lipids. We also have some peptide hormones, some protein hormones. So they're also used for regulation of cell metabolism. And then for motion, not just motion like we think of muscles, but actually even inside the cell, we have little microtubules of protein that move things around inside the cell. And then, of course, our muscles are filled with actin and myosin, which are proteins, little contractile proteins that let our muscles contract, and that's how our whole body moves. So amino acids, there are 20 different amino acids. Every one of the amino acids is the same here. So 20 amino acids all have this foundation, carbon, hydrogen, an acidic group. This COOH is an acidic group, an acid, that's why we say amino acids, and an amino group. Amino, when you see amino, think ammonia, means nitrogen containing. There's a nitrogen in there. So this is my basic structure for 20 different amino acids, and then they have this R group attached, the rest of the molecule. This is variable. This is where I get my 20 different amino acids. So same, all the way through. And then here's where I get my difference. So some of these have sulfur in them. But you see different combinations of carbon, hydrogen, oxygen, some nitrogen, and 2 with sulfur. So that's my difference. That's what gives me 20 different amino acids. And then I'm going to string these together in different ways. That's going to determine what their final shape is. So a protein can't function properly until it's in its final proper shape. If a protein doesn't have the correct shape, it can't do its job, or it's less effective at doing its job. So when a protein loses its shape, we say it's been denatured. And I can denature a protein, I can make it lose its shape by changing the pH of the environment it's in, by changing the temperature, can disrupt protein structure by exposing it to certain chemicals, because the shape of the protein is held together with hydrogen bonds. And so all these are able to disrupt those weak hydrogen bonds and mess up the shape of my protein. You've all seen a protein denature if you've ever cooked an egg. So an egg white is albumin. That is a protein. And it's clear when it's in its proper shape. When all those albumin molecules are in their proper shape, they line up so they are transparent. When I add heat, they lose their proper shape. Their structure falls apart and it turns white. So that is a protein denaturing. So proteins have 4 different levels of structure. My primary level is my chain of amino acids. Depending on, I've got 20 different amino acids. Think about this as being, the alphabet. I've got 26 possibilities. I put these together in all kind of different orders. So here I've got 20 different amino acids in a different order. And what happens is an amino acid here is going to say, oh, look, I'm going to have a little chemical reaction, or I'm going to form a bond with this one here. Oh, I can form a hydrogen bond here. Oh, I can form a hydrogen bond here. Depending on what the order of amino acids is, those hydrogen bonds might cause this big long chain to spiral or to pleat. So I could coil, or I could just get little bends in here. And which one of these happens depends on what my order of amino acids is. This is the secondary structure. So my secondary structure depends on the order of amino acids in my primary structure. Then I can form more hydrogen bonds. Oh, look here, we're going to form a hydrogen bond here, and this part of the molecule will form a hydrogen bond with this part. And so these shapes, either my helix, this is an alpha helix, a beta pleated sheet, these will twist back on each other into this three-dimensional shape that's very specific, because it will only twist into this correct shape if it has the right secondary shape. It won't have the right secondary shape unless it has the right primary order of amino acids. So that's my tertiary level of protein structure. At this point, it has a very specific shape, and this might be my protein. Some proteins are bigger molecules, and they'll take multiples of these tertiary structures and join them together. Hemoglobin is an example of this, and hemoglobin is 4 of these tertiary structures. One hemoglobin molecule can carry up to 1 billion oxygens, just to give you an idea of how much bigger we're talking when we're here at amino acids versus oxygen. And our blood cells are filled with hemoglobin molecules that are carrying, or each blood cell can carry up to 1 billion oxygen molecules because they're loaded with all these hemoglobins, each carrying an oxygen, 4 oxygens in its space. In this particular shape. So just to get back to the importance of this, with an example you might be familiar with, a blood disorder that that you might be familiar with is, sorry, had to cough, so I had to get a drink of water. So sickle cell anemia, you might be familiar with that. It's a blood disorder where the hemoglobin can't carry the right number of oxygens and the red blood cells are misshapen. Normally a red blood cell is a circle, a little concave circle, looks kind of like those red hot cinnamon hard candies. In sickle cell anemia, the red blood cells are sickle shaped, meaning they're just sort of like a low bowl shape. And what happens is those blood cells can't tumble and flow by each other in circulation. They stick together. And so there are clotting issues, there are circulatory issues, they're not carrying enough oxygen. So all sorts of secondary symptoms and problems because cells aren't getting the oxygen they need. It's this really, really complex disorder that has all sorts of symptoms related to this misshapen hemoglobin, is all due to one incorrect amino acid. So I can have my nice big long chain that's going to bend and twist and fold and join together. And if I get one wrong amino acid in one place, I just substitute one of these. So I'm going to have a substitution of, I think it's this, glutamine and valine. Oops, put the wrong one in. And that leads to an incorrect folding, which leads to an incorrect tertiary shape, which leads to a molecule that's shape is just off enough that when I fill up my red blood cells with it, my red blood cell is the wrong shape. And then I get this whole cascade of physiological impacts because of one improper amino acid. So that's how important these levels of amino acid order, these levels of structure for proteins are. And just an example that you might be familiar with. All right, nucleic acids, our last group. We'll push on through here. We're going to spend a lot more time on nucleic acids when we get to DNA, so I'll be fairly quick here. The monomers are nucleotides. I have two nucleic acids I'm interested in here. DNA, deoxyribonucleic acid. This is the genetic material that has all of the information a cell needs, a body needs, for every function it has to perform. So it has information on how it makes copies of itself so I can make new cells. But what it really carries when we talk about the genes, the directions, we say, oh, DNA is an information carrying molecule. It carries the information for how to put the right amino acids in the right order to build a protein, to build any of the proteins I need. So I'm going to build the enzymes I need to carry out chemical reactions to perform the functions of the body. That's where I get that information from. RNA, ribonucleic acid, has lots of different functions within a cell. It's also an information carrying molecule, and it's involved in protein synthesis. So this has all the information for how to build all the proteins a body might need. This is going to be one of those recipes for one protein. So we'll look more closely at that coming up. The structure of nucleotides, I've got a phosphate group attached to a pentose sugar. Pentose, it's a 5-carbon sugar. Remember, carbons can have anywhere from 3 to 7, or carbohydrates, sugars can have anywhere from 3 to 7 carbons. In my DNA and RNA, it's a pentose sugar, a 5-carbon sugar, and there's a nitrogen-containing base attached to that. So there are 5 nucleotides. That's all I have to know about for my nucleic acids, for my DNA and RNA. Those nucleotides can be adenine, guanine, cytosine, and thymine. And in RNA, same, adenine, guanine, cytosine, but I don't have thymine. I have uracil substituted for thymine. And then I stack together these nucleotides, these building blocks that look like this. This is my one building block. So this is a little more complex because it's got 3 parts: a phosphate, a 5-carbon sugar, a nitrogen-containing base. There's my nucleotide. And then I string them together to make DNA or RNA. So this is my monomer. And then I pile it together, one on top of the other, to make my nucleic acid. So my nucleotides, my 5 nucleotides, I have pyrimidines and purines. We'll go into this in a lot more detail when we get to DNA. I remember in DNA, we have cytosine and thymine, and adenine and guanine, not uracil. The way I remember which ones are the pyrimidines is they're the ones with the Y. So if it's got a Y, it's a pyrimidine. If not, it's a purine in DNA. And then I know in RNA, I don't have thymine. Uracil gets substituted in instead. So that's my other pyrimidine. The important thing to note here is my pyrimidines are single ring structures. My purines are wider. They're a double ring structure. And that's going to be important when we see how DNA is built and how it functions. So my structure for DNA and RNA, they both have a backbone. So their backbone of both DNA and RNA is an alternating sugar phosphate. So that 5-carbon sugar and phosphate attached to it, I'm going to stack all those one on top of each other, and that's going to be my backbone for my molecule. So RNA is usually predominantly a single-stranded molecule. DNA is a double-stranded. And by double-stranded, we mean it has two backbones. So it's like a ladder. It has two of those backbones connected by rungs. So DNA has two strands that are held together by hydrogen bonding between the nitrogen-containing bases. So my molecule has one of these stacked on top of another, stacked on top of another, and then a second row over here with the nitrogen-containing bases hydrogen bonded to one another. And the two strands twist a little bit, and they form a double helix. So here I have a sugar phosphate backbone. Here's a sugar phosphate backbone. So double-stranded, and then nitrogen-containing bases oriented inward and connected with these hydrogen bonds. So I get this spiral. The sugar in DNA is deoxyribose. That's where I get my deoxyribonucleic acid. Forgot an O in there. Then on either strand, so along one strand here, those nucleotides can be in any order, but the complementary strand, the other strand, has to have its complementary nitrogen-containing base. So there's a specific way these can pair up here in the middle. And it's always going to be, you see I used the analogy of a ladder. The backbones are parallel. So I always have to have the same distance across. And my purines and pyrimidines were different size. One was 1 ring structure wide, the other was 2. If I put 2 single rings, right, 2 of my purines together, this wouldn't be parallel. It would sort of narrow in here. If I put 2 of the double ring structures together, I'd get a bulge. They wouldn't be parallel. So the only way I can stay parallel is if I always have a purine with a pyrimidine. And the base pairing turns out that the number of purines always equals the number of pyrimidines because I always base pair one purine, an adenine, always pairs with thymine and only thymine. Cytosine, a pyrimidine, always pairs with guanine, a purine. So I always have this 1-to-1, 1 purine, 1 pyrimidine. And the reason I don't have adenine forming a pair with cytosine, even though that's a purine pyrimidine, or thymine with guanine, is because the number of hydrogen bonds that they can form. Adenine can form 2 hydrogen bonds. So can thymine. Cytosine and guanine can each form 3 hydrogen bonds. So if I tried to pair adenine with cytosine, what would happen was the cytosine would have this one additional place that needed to form a hydrogen bond, and it wouldn't have anywhere to go, so it would attach to some other part of this molecule and it would cause a kink or a break in the molecule. So the only way I can get my pairing is adenine to thymine, cytosine to guanine. So ATCG. So there's my hydrogen bonding for cytosine and guanine. I form 3. Adenine and thymine, I can only have 2, resulting in this shape. And I can describe this shape as a double-stranded molecule, the 2 backbone strands made of sugars and phosphates. And each of these has an attached nitrogen-containing base pair, my nitrogenous bases. The backbones run anti-parallel, and we'll talk more about that when we get to our unit on DNA. And my base pairing is complementary. These are my complements. So we'll talk more about this anti-parallel, but the example that I will give you is a road is anti-parallel. Both lanes, 2-lane road, got 2 lanes, they're parallel, but cars are running in opposite directions. So here, my sugars and phosphates are running in opposite directions. Ribonucleic acid, RNA, is a similar structure. The backbone are these sugar phosphates, the repeating units of sugar phosphate. The sugar in RNA is ribose. It's usually found as a single-stranded molecule, although it will sometimes double back on itself. So there's no base pairing, but it does have a lot of important functions. And remember, I don't have thymine in this molecule. I have uracil instead. So this just sort of summarizes and compares DNA and RNA. I have deoxyribose, ribose, adenine, guanine, thymine, and cytosine. Here my thymine is substituted out. Instead I have uracil. DNA is double-stranded with base pairing between the strands, and RNA is single-stranded. That helical twisting shape, DNA has it, RNA does not. My last nucleic acid that I'm going to mention is just a nucleotide. It's ATP, adenosine triphosphate. It's a nucleotide composed of adenine, so that's the nitrogenous base is adenine, and ribose is the sugar. So remember, my nucleotide is a 5-carbon sugar, ribose, a nitrogen-containing base, and a phosphate. ATP has 3 phosphates attached to it. This is the molecule used by the cell for energy. Right? It's the only way the cell can get energy, at least energy that it can use. So this is what we're going to spend a lot of time talking about when we look at cell respiration and metabolism, because we need energy to do work. So this is what my molecule looks like. There's my 5-carbon sugar. There is my ribose, my adenine, and 3 phosphates, adenosine triphosphate. And I'm going to release energy by breaking off that phosphate. So that is it for my biological macromolecules. Hope you all have a good weekend. Enjoy your holiday Monday. Use it wisely to rest and recover, and maybe for a little bit of review or catching up if you need that. All right. We'll see you all soon.